Alright, welcome to another Biology Homework Lab video, and now we're going to be looking at the parts of the microscope, and so let's get straight into it, shall we? Alright, so microscopes come in different types. We said that in previous videos. They can be light microscopes, they can be electron microscopes. And remember, if you do a bit of active recall, what were the different types of light microscopes? That's right, we broke them into simple and compound. Remember, we said simple is when they're using one lens, and compound was when you were using two or more lenses. And then we said that the compound microscopes can be different types: monocular, binocular, and stereoscopic, right? Well, here we have some examples of compound microscopes because they've got one lens up here and another lens down there. So there's two lenses that you use. This one here is a very simple one you see in very junior science labs, okay, where we're just using light from a light source. Could be just the light from the window, or it could be a lamp, and that light gets bounced by the mirror up. Whereas this one here's got a integrated light into the actual base of the microscope, so it's a more expensive. It's a compound microscope, but it's also a binocular. Here's got two lenses, two eyepieces there. Okay, so it's a binocular version. So we're going to look at the parts because they're similar between the two. So the parts of a microscope can be broadly categorized into three sections. The first one is those that do the magnifying, right? And so they're the lenses. So up the top here we have what are called eyepiece lens or an ocular lens. Ocular means eye. Okay, so these are our eyepiece lenses, and generally, if I just get a different color here, I'm going to get white actually because we've got a blue background. These ones here are 10 times, right? They magnify things 10 times. Okay, so that's what their magnification is. Now, with more expensive microscopes, you can put different ocular lenses in. You can take one ocular lens out and put another one in, so you can have 2 times, 10 times, 20 times. So they're more expensive ones, but you probably see in most junior science labs that the 10 times lens is what you're using. The second type of lens is located down here. These are called objective lenses, and they may differ in the number and quality, okay, and the type. But these are objective lenses, and so you can see these objective lenses here. They're sitting on a structure called a nose piece, and that nose piece rotates, so you can move those lenses around, so you can get different magnifications. And look, sometimes they're 10 times, then we have 20 times, and 40 times, for example. They may be 5 times over here in the simple one. Let's just get that back. So it might be 5 times, then 10 times, and then 20 times. Depends on the microscope quality that you've got. But they're the lenses, right? We have ocular, what we see, and then we have objective down at the bottom. All right, that's the same slide. There we go. All right, so we have the parts which illuminate now. So we've talked about the lenses. Now we're looking at parts which illuminate the specimen. That's what you're looking at, right? So the specimen goes there, right? Usually on a slide, okay? But what do we use to illuminate it? Well, here in this simple one, we've just got a mirror which can rotate backwards and forwards. So the light comes in, bounces off the mirror and goes up through the specimen, then it goes up through the lens, through that lens, and then into your eyeball, okay? This one over here has got an integrated light source, okay? On-off switch down the bottom. It's got an intensity slider there where you can change the intensity of the light source, so therefore you can be more sophisticated in how you look at your specimens. Then from the light source, it's got to go through two objects, right? The first object it goes through is called an iris diaphragm. Now, you've heard of an iris before, the iris in your eye, right? That is the colored part that you see when you look into someone's eye, and that's what dilates or contracts and changes the size of the pupil, the black part of your eye. So the iris opens and closes, opens and closes. When you're looking at a camera, inside a camera lens, they have the iris of the cameras with different blades that open and close, which changes the amount of light that goes into the camera, right? So here we have an iris that changes the amount of light that enters the actual specimen, right? So we can change the amount of light. Then once it goes past the iris diaphragm, it's got to get past what's known as the condenser. Now the condenser helps focus the light. It condenses, right? To condense something means to make more concentrated. right? When you're cooking, you might say, oh, I got to condense that. You add some condenser there. It's got to make it more concentrated. So here, in terms of light, to condense the light means to bring it together, to focus it a little bit onto the specimen, right? Once it's done that, then of course you can then see it with your lenses. So that's sort of the illuminating parts, right? Now we're looking at some mechanical parts here. In other words, to get the function of all these things and get them to work together. So you've got your body tube. This is just the section of tube here that allows light to go through from the objective lenses up until the eyepiece. And see, here's the body tube here. This one's more sophisticated because it's got prisms in there. Those prisms just allow the separation of the light, so you have two for each eye. We have the arm that helps in holding the microscope and transferring it from one location to another. You've got the base down the bottom here that's not labeled, that just supports the microscope. Okay, now we have these knobs here, and they're labeled coarse adjustment knob, but this has also got a fine adjustment knob there. Some microscopes have the coarse and the fine adjustment knob on top of each other. Now what do? Well, the coarse knob adjusts the height of the stage. So this is the stage, so let's write that in, the stage. Now what's the stage? It's just the place, it's just the location on which you place your specimen. So if we move this knob, this stage goes up and down, and that focuses the light. Okay, it's sort of a bit like if you've done with your camera, you hold your camera and someone moves backwards and forwards, then it's got to focus, doesn't it? The camera's got to readjust its focus, otherwise you go out of focus. So that's what happens here with the stage, right? That's the coarse one. It just means it moves the stage really fast. And then you have the fine adjustment knob, which is for very small movements of the stage. And so let's say you focus your specimen. If you move the fine focus knob, it goes out of focus too quickly. So then you use the fine focus, and it just moves the stage upwards and downwards a little bit to get fine precision focus. Okay, now we've talked about the stage. Okay, the place in which you put your specimen. It's held together by stage clips. Now with this simple microscope, they're just clips which hold the slide in place. You have to physically move it with your hands if you need to change its location. However, with our more expensive microscope here, you can see they've got a mechanism that has like little dials you can turn, and that helps move the slide in all different directions, so you can be more precise with your movements, right? You can imagine that if you're looking through here and you move with your hand, that's going to be a massive movement when you look through the microscope. So this is more precise. And we've talked about the fine adjustment knob, which is actually that one there. So that's mislabeled, that's the coarse one. All right, the fine one is just there. So that's it. That's looking at all the parts of the microscopes for this video. Make sure you've got these down in a table. You've got like the parts and then the functions. Some way that you can memorize all the different parts of these, because when you go to use them in the lab, you need to understand what they do. All right, I'll see you in another video.